Welcome everyone to this first day of SciLifeLab Outreach Week. And the title of this afternoon's program is Your Idea Ready for Drug Discovery? And my name is Lena Gustavsson. I'm the Director of Chemical Biology and Genome Engineering Platform. And together with Per Arvidsson, Director of Drug Discovery and Development Platform, I will be moderating this session. The program includes presentations of our two platforms, what services that are available, but we also, most importantly, have presentations from 3 of our users describing their experiences in using our platform. So a very special and warm welcome to Marika, Sonja, and Margit. Before we start the program, I will introduce SciLifeLab. So SciLifeLab is a national resource of unique technologies, expertise, and instrumentation, but also with a strong research community within life science research. The mission is to enable life science research that would otherwise not be possible. And you may also know that SciLifeLab is distributed all over Sweden. SciLifeLab as a concept is now more than 10 years old, and it was funded in 2010 as a research collaboration between 3 universities in Stockholm and also Uppsala University. But it was not until 2014 that SciLifeLab got the mission and funding to provide services as a national infrastructure. So one could say that SciLifeLab rests on 3 pillars: a strong research community, infrastructures with services, but also this 3rd pillar, which is the newly funded SciLifeLab and Wallenberg-funded national program on data-driven life science. And this is a major strategic initiative with 11 partners in Sweden. Today you will hear more about the infrastructures. And the infrastructures or the services that are available for users are organized in 10 platforms with different technologies, instrumentations, and expertise. And the platforms are organized to be able to address and enable research capabilities or research areas, either within one platform or together or integration of several platforms. And these capabilities or research areas, they span from precision medicine, pandemic preparedness, system biology, structure biology, or what you will hear more about today, drug discovery. And the drug discovery process is usually described along this type of axis with different steps, starting with target discovery identification phase, following in identification of a starting point that modulates your target of interest. And these modulators may be small molecules, antibodies, or other modalities. And then the project follow an optimization steps and before you enter the preclinical development stage. And finally, and hopefully, your project reaches the clinical studies stage. And our platforms try to support your projects or the research projects to progress along this axis. And typically, our platform supports the early phase of the drug discovery process. But of course, we also have other platforms at SciLifeLab that may support your project as well during this journey. And today, you will hear more about different examples of projects that have been using our platforms. And with that, I would like to start the program, but I would also like to say a few words that I will try to keep an eye on the time. And there will be some time for questions after each presentations. And then Per will help me monitoring if there are any questions, and also you can write questions in the chat, of course. So the next speaker will be my colleague in the platform, the co-director Bernhard Schmierer, who will present the chemical biology in genome engineering platform, capabilities, expertise, and technologies, and some examples. So please, Bernhard.
Thank you. So as Anna-Lena said, I'm co-director of the chemical biology and genome engineering platform, which is one of the two platforms that is presenting here mainly today. So the other one is the drug discovery and development platform. And I am heading CRISPR functional genomics, which is one of the 3 units of our platform. So the platform has 3 independent units. One is the Chemical Biology Consortium Sweden, of which Anna-Lena is the director at the KI node in Stockholm, and Eric Corell at the University of Umeå node. And recently, CBCS has actually received funding from Vetenskapsrådet, and they will establish new nodes also at Lund, Uppsala, Gothenburg, and Linköping universities. So my own unit, CRISPR functional genomics, is also based at Karolinska here at Biomedicum, and we are focusing more on genetic aspects using CRISPR. And the third unit is the chemical proteomics unit, also based in Biomedicum in Stockholm, and is headed by Massimiliano Gaetani. And as a platform, so the 3 units together, we have also a common mission, and that common mission is to turn phenotypic observation into mechanistic insight. But before I say more about the synergies and the platform capabilities, I will briefly introduce the technologies of the single units. And I start here with Chemical Biology Consortium Sweden. So CBCS, as it's also called, provides chemical compounds. So it enables medicinal chemistry. It supports assay development and miniaturization, as well as chemical screening. And it can profile compounds in various ways. So what can be done at the unit is to create, for example, small molecule tools for basic research. You can unravel biological function and mechanism by using known small molecules of drugs. For example, CBCS has access to the AstraZeneca compound collection. So there's a phenotypic compound collection that contains drugs with known targets or mechanisms. You can probe druggability or ligandability of target proteins with small molecules. Can identify and also optimize small molecules that modulate the phenotype of interest. So this is done by chemical screens, and then the optimization by medicinal chemistry. You can establish a structure-activity relationship by synthesizing and testing analogs of a hit compound, for example. And you can profile small molecules phenotypically and multidimensionally. And some of these dimensions are cell viability, cell type specificity, cell painting, transcriptomic readouts, proteomic readouts. Okay, so in summary, I think CBCS provides the whole package for essentially the initial steps of a drug discovery project. In contrast, my own facility, CRISPR functional genomics, is more, so it uses not chemical, but genetic tools. And we focus predominantly on high throughput applications of CRISPR, but we also do precision editing in cells with CRISPR. So our major focus are genome-wide CRISPR screens, all sorts of different modalities of these genome-wide CRISPR screens. It doesn't have to be genome-wide, it can also be a focused screen. We also use base editing CRISPR screens to introduce mutations and to do mutagenesis screens. And on the precision side, here it's relevant that with precision editing, you can create CRISPR reporter cell lines and disease-relevant model systems. So for the CRISPR savvy amongst you, have here in the toolbox what we have available. So we use regular Cas9, we use dead Cas9 fusions, we use other Cases such as Cas12a, Cas13d. We do screens by knockout, CRISPR inhibition, activation, base editing, and also single cell readout methods such as Perturb-seq, Crop-seq. And in the precision editing part, we can do gene knockouts, but also knock-ins of tags, point mutations. We use base editing, prime editing. So we have essentially the whole toolbox up and going. So what can you do with us? You can, as I said, create disease-relevant cellular models by doing some precision edits in your gene of interest, or you can create reporter cell lines for screening, for example, by knocking in a GFP or a luciferase. In the big screens, you can interrogate protein coding genes, but it's not limited to that. You can also interrogate long non-coding RNAs, microRNAs, even non-coding DNA elements such as enhancers, promoters, etc. This can be done both genome-wide or with focused libraries. So you can, with these applications, identify genomic loci, very generally speaking, that are regulating all sorts of biological processes. Examples for screens we have run is for viability, proliferation, cell differentiation, for drug response, for infection by viruses and bacteria, signal transduction, cell-cell interactions, etc. And you can also map small molecule target interactions by CRISPR-guided base editing scripts. So this is maybe more in the drug discovery direction. Finally, the third unit of the platform is chemical proteomics. So chemical proteomics focuses essentially on changes in the proteome, proteomic changes in response to small molecules. And the facility has a number of cutting-edge technologies that are actually developed in-house. And what can be done at this facility is to identify small molecule targets by deep profiling, 8,000 to 10,000 proteins. You can study the proteome regulation and proteome function, importantly at a multidimensional level. So, for example, protein expression, thermal stability, post-translational modifications, redox balance. You can identify small molecule targets by combining different orthogonal proteomic approaches. So these are also based on different physicochemical properties that can be exploited here. One can map binding and interaction sites, so binding sites of small molecules and compounds, but also interaction sites of, for example, antibodies, so epitope mapping. And one can narrow down targets of several small molecules in parallel. So the facility provides the possibility of multiplexing here. Okay, so this is essentially what the individual units can offer. As the platform, we are also interested of actually making use of synergies that arise from applying these genetic, chemical, and proteomic methods. And these synergies arise predominantly in the fields of target discovery and mode of action elucidation. So it is maybe not appreciated enough that identifying and validating targets of bioactive small molecules that elicits a certain phenotype in an assay is actually very challenging. And combining several approaches from different fields is basically needed to get anywhere close to a target. And here I think the platform can provide exactly these different angle approaches. A related point is the mode of action elucidation. So how does a small molecule elicit its effect? Which pathways does it affect? How does it interact physically with the target protein? And also, of course, importantly, are there any off targets? Is the effect really mediated through the presumed target protein, or are there any off targets that actually mediate the effect of the drug? So this is all very important to know. And our three units together, I think, provide quite a toolbox to approach these questions. And the goal essentially is, by performing these target discovery and mode of action elucidation studies, to ideally come up with a small molecule target pair. So this then can be fueled further into the drug discovery development platform, who can then try to develop this further. As an example of what the platform can do, this is a project that was run at Karolinska Institute by Nico Dantuma and the PhD student Tatiana Alvarez Giovannucci. So here they created, so the customer themselves, this is a project that went on for a very long time. So this took basically 10 years from beginning to end. The goal of the platform, of course, now is the platform is new. We're trying to accelerate this process a lot. So the way it worked here was that the customer themselves created a model system that is a ubiquitin intact with YFP, so with a fluorescent protein. And they were then basically looking in a phenotypic chemical screen for compounds that would inhibit proteasomal degradation of this reporter. The screen was run at CBCS. They screened about 23,000 small molecules, tested analogs, established structure-activity relationship, synthesized probes, and eventually then came up with a hit compound that, when cells were treated with this, blocked proteasomal degradation of the reporter. This was then changed by medicinal chemistry, improved into a more active analog. And at the same time, an inactive analog was created. So this is also an important thing to have, an active and an inactive analog, can study specificity properly. And this probe then, the active analog was then chemically modified to act as a probe for affinity-based mass spectrometry. And as you can see here, a number of hits were found that would interact. They were pulled down in mass spec with the probe. One of these hits was NQ01 that just showed up somewhere amongst others. So this shows very clearly that this approach alone is not enough to actually identify a target. So what was then done at CFG, so at my facility, was a genome-wide CRISPR knockout screen with the same reporter cell line. And here NQ01 came up as a top hit. So you can intersect the hits that you find with the affinity-based mass spec and the genetic method. And then, of course, you have a much higher confidence that this thing that you're looking at, NQ01, actually is a target. And in this case, it turns out that NQ01 is actually an enzyme that turns the compound into its active analog. So the compound CBK77 is turned by NQ01 into a ubiquitin-dependent protein degradation inhibitor. And this is important, an important drug for cancer treatments if it is developed further. The project has been published now 2021, and there's also follow-up funding from Novo Nordisk funding available for this. So it will be exciting to see where this actually goes. So as a summary here, the chemical biology and genome engineering platform consists of 3 independent units. The units can be engaged either individually or in combination, depending on what the project is. Individually, we offer a wide range of cutting-edge technologies in chemical biology, chemical proteomics, and in CRISPR-based functional genomics. We have strong synergies between the units in the focus areas of target discovery and mode of action elucidation. And our general aim, the unit and the whole platform, is to support the unraveling of biological function and mechanism, to facilitate and accelerate both basic and translational research, and to enable, in the end, high impact publications. So finally, contact details. We are, of course, happy to discuss your project, your questions, and we do that confidentially and free of charge anytime. Just send us an email. So there's Anna-Lena and Eric for CBCS, myself for CFG, and Massimiliano Gaetani for the chemical proteomics unit. And finally, I'd also like to say that we are hiring. So chemical proteomics is hiring an expert in mainly chemical proteomics and HDXMS. Myself, I'm currently looking for a research specialist in CRISPR screening, CRISPR functional genomics. And CBCS got substantial funding from VR and is looking for actually several people, both at the KI nodes, the Umeå node, and also at the new nodes in Uppsala and Gothenburg. For details, please look at their webpage, cbcs.sc. And with that, I'd like to thank you for your attention, and I'm going to take any questions if there are any. Thank you.
Thank you, Bernhard. So it's really exciting recruiting, so please keep an eye on our homepage. Maybe there is something of interest, but also to contact us for collaborations. Are there any questions, Per?
Yes, please feel free to use the chat to ask your questions. I will try to read them in order to save some time. So I have one question from Carl here, and he wondered if you consider nowadays RNA routinely as a target for bioactive small molecules.
I'm going to pass that right back to you, Per, actually.
It's a very interesting development to target RNAs directly with small molecules. And this was not a question about oligonucleotides, as we will return to. We will have an opportunity to offer, as Kristian will say, oligonucleotides also from the fall as a new modality. But in this question, it's about small molecules targeting RNA, and that is a very hot and upcoming area. We have not yet been in contact with such a project from our side. I don't know if you have, Anna-Lena, on the screening.
No, not that I can remember right now. We would need to discuss that more details. I'm not aware if there are any special requirements on the small molecule side to address that, but possibly it would be the assay setup that would be interesting to discuss further.
Yeah, there are some known chemotypes which are often seen for RNA binding, but so I guess it's also one little bit depending on what kind of library, if you could do a pre-selection in the chemical space.
Mm. But that is definitely something we could look into. I ask you, Carl, to contact us after that meeting. We can have a discussion and see what we have in our compound collection, for example, if there are certain chemotypes that might be of interest, for sure.
Yeah, he thanks here. I don't see any other question right now. But please, if you have other questions to Bernhard, meanwhile, just type them in and I will pick them up at the Q&A session afterwards.
So thank you. So let's continue. We are a few minutes ahead of the program, but so by that, I would like to introduce the next speaker, who is Kristian Sandborg, who is director at the Drug Discovery and Development Platform. Kristian will present the drug discovery and development platform capabilities, expertise, technologies, and also some examples, I believe. So please, Kristian.
Thank you. I'll try to make this presentation ready. Do you hear me? Okay, good. So let's see now.
Now you stopped sharing, Kristian.
Yes, I had to
Okay.
do that. Trying to. Finally.
Now you the presentation.
Is it okay?
Now you're on.
You have a very.
Now it's perfect.
Perfect. Good. All right. Then I start. If I find a laser pointer as well. There we go. Yeah. All right. So my task here is to make a brief overview of the drug discovery and development platform. I'm the co-director of the platform, placed it at Uppsala University. So what I will describe to you is capabilities, expertise, technology, and one example of the platform. And we are placed at 6 different universities here in Sweden. So we are quite distributed between the sites. So the objectives for DDD, our existence really depends on the struggles to span this valley of death. That is research done at academic settings and how to apply that in clinical use, and then commercialization resources are really important there. And one of our main objectives is actually to do project scouting in the universities to find the best possible drug discovery projects that we can support. And what we can do is to give intellectual and wet lab support. And importantly, it's not just small molecules that we are working with. We have also since the start 2014 been working with antibody therapeutics as well as was exemplified before. This year we are actually starting up in Gothenburg operations for developing therapeutic oligonucleotides in the OligoNova initiative. But we are also working with what we call new modalities, that is totally novel therapeutic agents that cannot really be fitted into any of these categories above. This year, there is an opportunity now, I would say, to enter the portfolio of DDD, since we are launching 5 to 10 new projects this year. So I will come back to that. So capabilities within DDD. We are focused on two activities. One is to support the development or identification of new drugs. The other is to support new technologies that are of interest for drug discovery research at the universities. And we do that in different ways. And for the technology areas, I will not come back to that so much, but we have been working with the DNA encoding libraries. We have been working with artificial intelligence enabled systems. We are active in developing PROTACs, for instance, and massive virtual screening. Antibody therapeutics, we have our own phage display libraries, but we have also been working with bispecific monoclonal antibodies. For the future, we have focus areas. We want to dig in more to machine learning for drug discovery, and as you understand, therapeutic oligonucleotides. And we continue to work with display and selection technologies like phage display and other technologies. But we also have an interest in proximity-induced agents and protein degraders. So the road to DDD. As you understand, the minimal requirements to enter into DDD for a drug discovery projects is that you need to have a therapeutic ID to present to us. But the other very important thing is, as Bernhard alluded to, is to really identify the target that we are going to work with. All our technologies are actually target-based. And a third requirement is that since we are no experts in the target biology per se, we are really dependent on that you can provide the models and the target biology expertise to guide us into the right type of compounds that we are developing. So the users for us mostly are academic scientists in the universities, but it could also be in industrial partners and innovation offices, etc. The first step is to make contact, and you can simply email to this address here or find more information on the web page. But we have a first meeting or a consultation meeting or several meetings actually, usually. And in these meetings, you will get a professional second opinion on the concept and the potential to actually launch a drug discovery project. We can also help you with the analysis of the competitive position and give you an initial technical assessment of your project. And this is under confidentiality, and as always at SciLifeLab, we do not take any ownership of your project, so it's totally safe for you. It's important also to say that we can only support approximately 20% every fifth project with our own resources, and there are different flavors for that here. So most of the projects we all actually redirect to other players in the field. Could be many times other SciLifeLab units like CBCS, but also Testa Center, Zeros Industry, etc. Could be different. And this is an overview of how we look like today, year 2022. We are more than 40 employed people at DDD. And we have 11 different units or facilities. And they work with different parts in the drug discovery process. We have one important facility working with how drugs are taken up into the body and metabolized and distributed and secreted finally from the body. We also have a team working with the product profiling and drug safety assessment parts of drug discovery projects. 2 teams working with protein expression and characterization. 1 team very much focused on antibody therapeutics, which is placed both in Stockholm and Lund. We have 4 teams working with assays, different aspects, both biochemical and biophysical, and more cell-based assays. And then we have actually 3 teams working with chemistry and very close collaboration with the CBCS and their compound center. We have a national platform steering group that have responsibility for project prioritization, and we have an international platform advisory board that has helps us with the long-term strategic orientation of the platform. And I will not go into details here, but this is an overview, as I said. And I'm just saying that also we are hiring in Gothenburg chemistry and biology for oligonucleotide therapeutics. So look out for these positions. The portfolio overview right now in January is as you see here, DDD programs. That is our most favorable projects when we add resources from all of our facilities to progress your idea up to the stage when you can have investors supporting the program in the development phase. And it's divided in small molecules, antibodies, oligonucleotides, and new modalities. And currently, we have about 50% division between small molecules and antibodies. And as I said to you, we will have calls out now for antibodies and oligonucleotides, and maybe also small molecules later this year. We also have the opportunity to work in collaborative projects, and one example here is Enable2 projects, working with antibiotics projects, where we are supporting the lead generation phase in these projects. Another is focused on technology development in technologies that are of interest to us. But we also have the possibility to support very specific activities, like in this case, antibody binders for the IMI project Open, or bioanalysis in the conception project. And in addition to that, we are supporting smaller activities up to 2 weeks work in the labs, about 40 projects per year. Output from SciLifeLab, as an example, last year we helped to support 3 new biotechs, leaving us after approximately, in average, 4 years at the platform. We also noted that the two of our older projects that left us 2019 and 2020 actually were listed at NASDAQ. We started this support to enable 2 and antibiotic research. We started the work together with the Open. And we also were involved in pandemic preparedness and different projects for SARS-CoV-2. Am I late, Anna-Lena?
Yes.
So this is just to show you then that one example of the projects that left us last year, it was Sara Mangsbo. And it's a nice illustration that you need to come to us with a target ID and a defined target, which she did. And she also brought with her the target biology expertise. What we did was to actually design the therapeutic in this bifunctional antibody, and eventually hand it over, helped her to hand it over to the company, Strike Pharma AB. So I'm sorry to break there, but I'm open to take questions.
So thank you, Kristian. Is there any quick question for Kristian? Otherwise, we save more questions for the discussions, perhaps.
There is none right now, so I will hold as well until the discussion here.
Yeah, that we hold it for the discussion. Thank you. Thank you, Kristian. So now it's time for the final presentation by Margit Mahlapuu from University of Gothenburg. And she will talk about new treatment in NASH and her infrastructure experiences. Please, Margot, welcome.
Thank you so much. Do you see my presentation?
Yes.
Perfect. So I will talk about my infrastructure experience, and our project is within the area of metabolic liver disease, NASH. So I will start with a short recap of NASH pathophysiology. And the biggest risk factor for our healthy liver in terms of NASH is obesity. And since obesity is increasing, also the prevalence of NASH is rapidly increasing. In connection to overweight, our liver collects lipids, and this is termed as NAFLD, or non-alcoholic fatty liver disease. And in certain amount of patients, which is quite significant, as you see here, liver also develops inflammation and fibrosis, and this is determined as NASH, or non-alcoholic steatohepatitis. And importantly, if NAFLD is clinically usually silent, while NASH patients have high risk to progress into cirrhosis and hepatocellular carcinoma, or HCC. And HCC is the second most prevalent cancer. It's the only cancer which shows constantly increasing prevalence and mortality rates. So NASH is a very serious disease. And correspondingly, NASH has been defined by both EMA and FDA as one of the major challenges in healthcare. Partly it's due to high prevalence, and partly we lack treatments. It's difficult to say what is the prevalence of NASH in general population, because it's only diagnosed by liver biopsy. But in high-risk populations, such as people with obesity or type 2 diabetes, up to 30% are considered to have NASH. There are no drugs approved for treatments. The only option for people living with advanced NASH is liver transplantation. The most advanced drugs in pipeline are OCA and olifibranor. They have recently disclosed results, intermediate results of phase 3 trials. And unfortunately, these results were not too promising and did not provide basis for approval. So the NASH market is huge, and there is very pronounced medical need. And our objective here is to develop small molecules against novel kinase targets for NASH therapy. And specific objective for us is to bring the project to the stage, as we have discussed earlier today, which enables to pass over to commercial actors who can bring it closer to the clinic. And for this, we aim to reach technical verification, meaning non-clinical efficacy and safety based on current guidelines, and to secure IPR. And before I talk about how I have used infrastructure, both national and EU perspective, to reach these objectives, I would just like to convince you that we really have attractive targets. So here is some science behind our idea. So first we discovered that there was a very strong correlation between the expression of our targets and NASH severity in human cohorts. So as you see here on the lower right panel, so NASH is characterized histologically by these three parameters: steatosis, inflammation, and ballooning in the liver. And you see that there is our kinase has positive correlation to the severity of NASH. This is, of course, only correlation. It doesn't tell us anything about causative connection. So we silenced the kinase in human hepatocytes, and we saw that we can efficiently reduce ectopic lipid storage within hepatocytes because we can increase beta oxidation and lipid secretion and repress synthesis. And correspondingly, we can also reduce oxidative and ER stress, which are important cornerstone in molecular pathogenesis of NASH. And finally, we have proof of principle in mouse models. If we silence the kinase genetically or by antisense treatment, we get less liver lipids, less hepatic inflammation, stellate cell activation, fibrosis, and hepatocellular damage, which is again meaning that we have less, we can protect against NASH development. And when we overexpress the kinase, we get the opposite result. So for us, it was very clear that it's an interesting target. We would like to have pharmacological inhibitors, but my team is within molecular metabolism, pharmacodynamics. So we have no chemistry competence. So we also realized that we will not be able to develop these inhibitors ourselves. And to find this missing piece in the puzzle, we turned at the time to CBCS at Science for Life laboratory. And first of all, they developed an assay for us, which was compatible to 384 format. And at the time, they also performed a smaller screen of approximately 20,000 compounds. Unfortunately, at that time point, no promising hits were identified in the library. And then CBCS encouraged us to turn to ELF, which had access to a larger library. So ELF is an European lead factory. And the idea of ELF is that academic groups, but also SMEs can present interesting biology with innovative targets in an application process. And ELF is comprised of compound collection of more than 500,000 compounds and a screening center which provides industry standard HTS. So basically what PI has to do is to come in with application, and then the applications are reviewed based on medical need, competition, trackability, etc. But I also want to stress that one important part here is that in the application, one also has to present assay and to really show that it has an acceptable signal to noise ratio to be compatible to HDS format. And again, without this input, which we got from CBCS and later on also from DDD platform, we would not have been able to make a successful application because we don't have equipment for even this large scale. So we would not have been able to do it. So we are really grateful for this national platform. So some more slides about about European lead factories. That the library is then put together partly from private partners and partly from public partners. And as you see here, most of the compounds come from private contribution. today, they have then more than 500,000 compound libraries. They have screened more than 200 targets and returned to PIs more than 7,500 hits in total. So by the legal framework, each PI gets about 50 compounds per screen. And when it comes to therapeutic areas, they do all diseases with some overrepresentation in oncology and CNS. So I have around 2 screens so far at HTS, and here is one of the screens. So if this is small molecule 4D map from basic science to market, then I'm currently operating in this small right upper corner of HTS. And in this case, ELF made a screen on 530 compounds. They selected more than 10,000 to go through deselection and orthogonal assay. They made 8-point dose response curve for more than 4,000 compounds and 1,000 compounds, which are all rather potent, as you see here. They were subjected to tremor shift assay. And currently, CBCS ELF is at this final stage where they synthesize selected compounds and perform confirmatory and certain quite small scale selectivity assay. And then this results shortly in QHL, which is defined as qualified hit list of about 50 compounds, which are returned to me as a PI. Now, the next step for me is that with these hit compounds, I still cannot do much because I don't know anything about bioavailability. So to make even mechanism of action studies, I have to develop lead compounds, which again takes quite much effort and investment to optimize both efficacy and selectivity, but also solubility, ADME properties, stability. And now at this stage, I have been fortunate to be accepted to SciLifeLab platform. So the aim is that TDD platform will take the hits and combine in silico modeling and in vitro screening to make sure that we will eventually come to the lead compounds. So basically QHL will be taken over by SciLifeLab for improved hits, and SciLifeLab will also perform hopefully in vitro proof of principle and in vitro DMPK. And then the compounds are returned to me, and in my lab I can perform in vitro pharmacokinetics, and we have also animal models set up, disease models, so we can perform in vivo proof of principle. And I think the dialogue with SciLifeLab has gone very well, and I want to say that it's not like that we would handle lower compounds at this stage, but SciLifeLab has been involved during the whole discussion with ELF, partly making sure that I have an assay which is considered eligible, but partly also when there are chemistry questions coming up during the screening or downstream analysis process, then SciLifeLab has supported me in responding. And also we have, or SciLifeLab has already performed the protein expression and crystallization so that we're in good position to start without any delay as soon as QHL will be released. So as we also mentioned before, that it's a very competitive area. Of course, NASH is so severe disease, we all keep fingers crossed for our competitors, but we still also want to be successful. So we believe that what we try to do is to have a project team which really represents all the competencies from science to exit. So I represent competence in basic metabolic research and drug discovery. We have Per Arvidsson, who is chemistry expert, Hans-Ulrich Marschall, who is a liver clinician. He has been PI in a number of metabolic liver disease clinical trials. Lona Fletcher at KO Ventures for business development commercialization, and Anders Bergman Profora is our IP responsible. So we hope that we cover the main competence is to bring the project to verification stage, at least. So what has been impact for this infrastructure experience from my part? And I think that, first of all, I would never have been able to develop any pharmacological inhibitors in my own, because we lack chemistry in my lab and we lack equipment to do this sort of screenings, or the libraries, of course. So we have been fully dependent that there are these type of infrastructures which are accessible. And ultimately, I hope to use these new tools for mode of action studies. What we do now is, for example, genetic modification in animals or SCR experiments in cells. And of course, if I have access to the compounds, I can work much faster, and I can also ask questions with much higher precision compared to genetic approaches, for example. Then we are, of course, very interested to contribute to this solving the clinical problem. So we hope to develop these compounds further towards clinical applications. It has been also interesting learning moment, both for me and my team, in terms of chemistry, but also, for example, legal competence. The user agreements can be quite extensive, especially with ELF. And there I really recommend that even if I myself find it, for example, quite challenging, I think it's very useful to really invest the time which is needed as an academic PI to fully understand the rights and responsibilities, because if we don't do it, the consequences can be very severe. And of course, I hope that when we have secured IP, that I will be able to publish these excellent studies which have been done by all these infrastructure components, and also that I will be able to share these compounds with other academic users, so that we can together advance this field and hopefully solve this clinical issue at some point. Yes, so that was my slides which I wanted to share. So I stop here.
Thank you very much, Margit, for a very interesting journey, and also illustrating that CBCS compound collection is not always enough. So you can complement by screening a larger set of compounds, because screening small molecules is a numbers game. And also illustrating nicely that the hit is not the drug, but you need some optimization and further development, monitoring the bioavailability parameters as well. So Per, do we have any questions?
I have some specific question here to Margit, and before we can open the kind of the panel discussion. And is also here is asking if you have tried lipids as bioactive small molecule targets. And I assume the question probably refers to if you have looked at lipid pathways, I guess, and not lipids themselves.
Mm-hmm. No, we have not. Our aim has been to find inhibitors which hit specifically our kinase. Then we know that the kinase, when inactivated, has a general impact both on lipid catabolism and lipid anabolism within hepatocellular cells. But we also know that it, for example, acts on autophagy and on oxidative and ER stress. And we don't believe that it is necessarily only through reduced lipid storage, but there might be parallel pathways which hit these NASH cornerstones directly. So we have not tried to reach or to replicate or to mimic the effect of kinase inhibition through targeting lipid pathways. We have tried to find specific inhibitors to the kinase.
Yeah. Then we have another question from Stefan. This is more, I think this is directed to CBCS. Maybe when Bernhard discussed the ubiquitin project, the screen was done with ubiquitin green fluorescent protein. And the question here refers to if it's such a large protein, looking at the effect of a small protein like ubiquitin, would it be possible to have a smaller organic dye? Possibly something you could put in if you use a non-canonical amino acid approach.
So can I, Anna-Lena,
Yes,
because I'm
Bernhard.
not quite sure whether you mean the chemical screen that was performed or whether it was the CRISPR screen, but it is definitely for the CRISPR screen, a different method is possible. So the question is basically, if you do another approach, you might have to fix, because you do some staining, like for example, halotech knock-in, and the click chemistry needs to be fixed afterwards, but that is no problem, so that can be done. And what I know from this screen, from the chemical screen, so this was an extremely clear readout. So basically, it was black everywhere, unless in the wells where proteasome was inhibited and everything lit up bright green. So I don't think that in this case it was a problem, but of course there's generally a consideration whether you want to have a GFP or not, and other methods are clearly possible. Yeah.
Okay, I don't see any more specific questions right now, so I suggest, we could kind of switch to asking everyone to either raise their hand or use the chat and type in some questions to any one of the speakers, or more of general nature. It would be good to get a discussion going. I can, now everyone is there, at least I can start just asking some more general question while you start to use the chat and ask some questions. Perhaps I wanted to start and ask a little bit with Margit. You used international infrastructure kind of in the start to get the project started in a way, and Marika, you used another infrastructure like the Testa Center at the end of your project. I was wondering if you could comment a little bit about the interactions between SciLifeLab and these other infrastructures, being them either European or national. So specifically, Margit, what kind of the pros and cons of screening the large European lead factory and that experience as compared to using the screen in-house when it comes to cost and availability and so on?
I think the upside of using ELF in my case was that they had very large library. I think first time they screened 390,000, another time 500,000 plus. And it seemed like, even if I have kinase targets, it seemed like that my kinase is not like a typical inhibitors. So I think that gave I hope it gave us a good starting point to go on with optimization to SciLifeLab. Because ELF's business model does not include any hit to lead activities any longer, at least. What was the downside of using ELF is that I think that it is really very extensive legal network. It takes also considerable time and money because you can't handle it yourself. You have to have a lawyer to support you. And also it's a milestone-based project, so that it's not no strings attached, but you have to, when you go on towards the clinic, you have a quite heavy milestone burden in terms of money, and that might negatively impact your possibility to commercialize your hits, because early targets value can be sensitive.
Okay, very good clarification, and also I think you made a very good point about that you need the local facilities in order to demonstrate to the European ones that you actually have an assay that is possible. Then I turn the question to Marika about your experience at the kind of the backside and with Testa Center. Is there something you would like to see developing further in that interaction and the pros and cons of?
Yeah, So for us, I mean, this was really, I think, the first time that we did this direct tech transfer from SciLifeLab to Testa Center, because there usually there was previously a gap there. And I think for us it worked quite well in the sense that we need, in this kind of in-between phase, we need to validate that we can do have a stable cell line, and that we can have a stably transduced, stably produced antibody. And we need a lot of material for these toxicity studies that we are doing. And I calculated that if we had to order this material, this 1 gram of antibody that we produced at Testa Center, that would cost almost 1 million Swedish crowns for us to buy. So in that sense, this was very valuable for us, since we could kind of just test drive the whole scheme to see that, you know, it works, we can upscale the production and so on. But if it would have been possible to use this stable cell line further, to use this one and take it into a GMP production, that would be even more valuable. I mean, this was absolutely valuable, and we have good use for this. But since we are looking into ways now to produce this in a more GMP setting and so on, of course, you can see since some of the work has already been done, but we have to start from scratch again with doing the stable production at the CMO instead. So if it would be possible to in the future also, you know, kind of use the cell line, the stable cell line that we already have, and to continue with this production, you know, that would be worth a lot of both time and money.
Okay, good. So I wanted to return to the funding question in a bit. You talked about funding here. So no one of you commented on what is the actual cost of running a project, either at the CBG or the DDD platform. Could Bernhard and Kristian comment a little bit on what's the cost model for the users, and maybe then from the PIs, their experience in finding that funding?
Yeah, I can start quickly. I mean, I have to speak mainly for my own unit, CRISPR functional genomics. So we have, of course, like every SciLifeLab platform, I think we are providing a subsidized service. So actually we put money into each project that we run. And I can give a ballpark of a cost, a genome-wide CRISPR screen. It depends on how many samples there are, how complex the screen is, but roughly 150,000 crowns. That includes basically everything, including the next generation sequencing and also the data analysis. So this is where we roughly are with these things. So most projects are somewhere between 100 and a maximum of 300,000 crowns. And then I think I pass on to Anna-Lena, because I'm not sure what CBCS price levels are.
Right, so it very much depends on the assay throughput and the complexity of the assay. But a typical target-based screen, like a kinase, where you screen the full primary screening collection of 50,000 compounds, it would cost somewhere between 200 and 300,000 crowns. And that includes the compounds, the follow-up material, some initial chemistry that evaluate the hits, and also, you know, single point screening, more conformational screens, counter screens, and full dose response screens, and also testing of some analogs and resynthesis.
Yeah, I can comment from DDD. We follow the policies at SciLifeLab, of course, and you, as an academic scientist, you only need to pay for the reagents cost. We actually do not have any budget at DDD for reagents and service of instruments. So we are actually totally dependent on you to get user fees for that. It very much depend on the nature of the project and where you are, how advanced the project is, the cost for running a project at DDD. It spans actually from nothing to up to, I think, the highest cost for a single project have been 700,000 for 1 year. right now. But on average, I would say that maybe 300,000 crowns per year. But the most importantly, you need to prepare for the cost that comes when you're testing active compounds in animal models, and that is part of the budget you have for your internal project and research project. You need to get funding, research funding for these expensive animal, mostly animal testing, or if you have advanced cell models. I would say that is probably the most costly part for that. And I'm not sure if you agree with that, Marika and Sonja, etc.
I wanted to hear the PI's comment about the difficulty of finding the funding to start with for the kind of the early activities, and also would like to know if working with the infrastructure have helped you secure that funding, because we know that it can be a challenge to kind of separate like purely academic funding for funding which goes into more translational research like drug discovery. So if you could briefly comment on that from your end, would be great. Sonja, maybe start.
Okay, yes, thank you. Well, I think you have very reasonable prices. It's never been a burden for us to to pay the bills for you. It's been okay. But then we never carried out animal experiments. So, and I think that's something you're starting to provide, is, if I understand correctly, or?
No, we do bioanalysis and support, but we don't have facilities, and we can't keep all the. Kristian wanted to comment.
Yes.
Yeah, we can actually do some PK study. We do internally, but we don't have the capacity as Per said to provide that on a regular manner.
Mm-hmm. Yes. So yeah. So that's the most expensive part. Then when it comes to getting more funding, of course, well, I hope that I always mention that we've managed to get very potent molecules in our work, and I hope that has helped getting the funding we need to carry on. So I assume it does help, of course. Yes.
Would Margit or Marika like to comment on that?
I can comment on that. It has definitely helped me because we applied first to Vinnova and this SciLife project when we still had the commercial antibody that we produced together with a company, when we were still hoping that it would be possible to convince them to take this to the clinic. So we applied then first to Vinnova. And then we were rejected with the explanation that we had a very unclear IP situation, which was very true. So the second time when we applied, that's when we had already gotten it approved as a SciLifeLab project, and we had our first hits and so on. And that's when we got the funding. So I think this was quite implemental, that they really looked into that, we had the rights to the antibody and that the IP situation was okay. So I don't think we would have gotten the Vinnova grant if we hadn't done this through SciLifeLab.
I have a question from Alexandro here. He wonders what kind of academic funds are okay to pay for the services at the infrastructures.
So far, I haven't met one grant that I couldn't use for SciLifeLab actually, and I've been using the Swedish Cancer Society and Vinnova.
Really. It's a matter of who gives the money. They usually have some strings attached, and you need to read that. But from our point of view, you can use any funding you want from academic funding you want.
And as Alexandra asked, VR is very, very okay.
Yeah.
Can
I also wanted to add that there's a credibility associated with the SciLifeLab CBCS and DDDP that helps you to deal with companies later on. So there's a trust, and I think that's very, very helpful too.
Yeah. Margit raised her hand here as well. So.
Yeah. I also wanted to comment when it comes to funding agencies, that even agencies like ALF or Cancer Foundation, they are of course interested to see that the project can be brought closer to the clinic, and I would not stand credible to say that I myself develop small molecule inhibitors. They would also understand that. So it's very helpful to show that one has a framework in place which can bring the chemistry forward. So it has helped me to get even these traditionally academic grants.
Okay, thanks. There are also more questions in the chat here. Olle here is thanking you for a great day and excellent presentation. And he wanted to have a question related to the kind of the competitiveness of the academic infrastructures like the CBG and the DDD when it comes to keeping pace with the competition. So he would like to know if you could elaborate on the difficult balance and which direction should the two platforms go, or where is the Swedish strength and niche, and how would you move more quickly? How could you move more quickly? Kristian is raising his hand.
Yeah, it's a very good observation, Ole, really, and it's true. When we choose the projects, select projects that can run at DDD, one important factor is, of course, competition. But also, I would say that we favor novelty over validity of targets, because valid targets are already very much worked on by big pharma. So novelty is actually a very important factor. You can work on your novel target in your group for a period. The other is actually new technologies and platform technologies, actually, to find a project that can be used for different approaches. And I'm sorry that I couldn't describe had more time to describe Sara Mangsbo's project, but that is a very nice example of a technology platform, which also, for instance, Marika's radiotherapy could be an example of. So that is important. But I think we need to work a lot about novel ideas to invent new therapeutic modalities and stay on top of that.
I know we are running at the end of here, but I have one related question in the chat as well, and that relates to if there is an interest from any of the two platforms actually to test and validate and develop more complex in vitro models, such as microfluidics-based system, organ-on-a-chips and organoids and so on, and how that fits into the development. If Anna-Lena or Kristian or someone wants to.
Yeah, maybe I can start. CBG is definitely interested in such more complicated and very much near to patient complex models. So in some cases, we have technical development projects together with future users to develop the models and then to have them set up as a more service-based technology, for example. So yes, we are very much welcome to this more complex model system.
That would also, Kristian?
Just a short comment. Yes, of course, we are extremely passionate and interested in these more complex models. The problem we see in harboring that type of technology in the platform is that they are very much designed on specific projects, and we cannot hold all technologies. So we prefer to collaborate with other SciLifeLab platforms, facilities, or other units in Sweden together with that. What we can provide is, of course, a test bed to test how well your technology, your organoids or whatever, actually behaves when you have drugs, and how they can be used for screening. And I would also say that we are not really sure how the facilities for oligonucleotides in Gothenburg will be set up. It could possibly be that we need more advanced models there. That's yet to be understood.
Okay. I do have one more question from the audience. Then I have also some other more general, but I don't know, Anna-Lena, if you want us to close the...
Maybe we should have, maybe Margit has been raising her hand for some time. Is the
Sorry, it's an old hand.
It's an old hand. Good. So yeah, Per, maybe some final comments.
Well, it's one final question here from Isuzu, who was wondering about the phenotypic assays and how do you overcome the challenge of linking phenotype to genotype, since the two do not always correlate at the initial stage, especially for unknown variants or rare variants.
Yeah, I mean, if you have the phenotypic assays, you could identify the small molecules first and then combine that with Bernhard's studies, for example, to identify if it's possible. But I guess it needs to be done stepwise. Bernhard, what do you think?
Yeah, I would agree with that. This is, of course, a challenge. But in CRISPR is actually not, in a CRISPR screen, a phenotypic CRISPR screen is not that much of a challenge because we know what the perturbation is that we introduce, and we can read that back out again. So I'm actually not entirely sure what you mean with the question.
But we asked the user to contact us in more details on your project. Maybe we could figure out something.
Yeah, exactly.
Some ideas.
Yeah, I had also some other questions related to that, with what you would do differently in the project, for instance, now when you are combined, but I think that will also depend on the project. You will be able to move faster than the example on the Nicodantuma project there now, when you have the integration opportunity between the different technologies, and also the chemical proteomics, which we have not touched so much on, is more becoming an unavailable resource, which it wasn't just a few years ago.
Exactly.
Okay, I think we covered most of the user questions, which is the most important thing.
Yes, and I think we are some minutes over time, actually. I think this has been a very, very interesting 2 hours to hear more from our users, to hear the full story from both two type of modalities, small molecules and antibodies. And I guess in the future, we look forward to hear more about other modalities like oligonovas and that initiative as well. And we are very grateful for Margit, Sonja, and Marika to be able to take their time to come here. And also thank the audience for attending this outreach. I'm very happy to have, I think I noted, 150 people in the audience. So thanks a lot.
Yes, very much so. Thank you very much all for joining and for presenting.
Thank you.
Okay. Thank you.
It's great to see the progress by SciLifeLabs to the CBCS and the DDDP. I think, yeah, there's been a lot of new technologies coming in.
Yeah.
Yeah.
Good.
Thank you.